Okay, so we've covered one of the functions of DNA, and that's the ability of DNA to replicate itself, and that's how it functions in heredity, to be passed on to new cells and new individuals from parents to offspring, for example. But there's also the other function of DNA that is considered the central dogma of biology, because it's so fundamental to how all life on this planet operates. All life on this planet has DNA, and that DNA is transcribed and translated into proteins. And that's basically the central dogma. So a gene is a sequence of DNA that codes for a protein. But there are genes that also code for functional RNAs. In other words, the RNA that is transcribed from DNA can function all by itself. And we were discovering more and more different RNAs that carry out their own functions. Phenotype is the individual's observable trait resulting from its genotype. So its genotype are the genes that it has encoded in its DNA. And the phenotype is the expression of those genes, the outward appearance or the ability or the behavior that can be observed and described. Gene expression is the overall process that begins with the transcription of DNA sequence into an RNA sequence that's translated into an amino acid sequence of a functional polypeptide or protein that results in a particular phenotype. That's a mouthful, but notice sequence. It's all about the sequence. So the sequence, it ultimately starts with the sequence of nucleotides in DNA, and it ends with a phenotype, a certain function being carried out in a living thing. So gene expression is everything from gene to phenotype, including everything in between. How do we go from gene to phenotype? That's gene expression. And it encompasses the entirety of the central dogma of biology. So like I said, it's considered the central dogma because it's so central to how all life operates. The role of RNA in the process is like a middleman. So you'll notice it's right here in translation. And it's actually the RNA that is physically stringing amino acids together to form the polypeptide and protein. So they do the work. The RNAs do the work. And they carry the information. So like DNA, and that's the focus, that's what we're focusing on right now, is RNA. And how's RNA different than DNA? So like DNA, RNA is a nucleic acid, and it's similar to DNA in its variable sequence. In other words, just like DNA can have the nucleotides that make up DNA can be in any sequence, the nucleotides that make up RNA can be in any sequence. The sequence is variable. Therefore, it can carry information, just like DNA can carry information. And in the case of messenger RNA, it does carry information, and also transfer RNA. In messenger RNA, that would be the codons, and in transfer RNA, it would be the anticodons. Those are information. Here we're looking at just a very little piece of RNA, but all the RNAs, even though this is specifically messenger RNA, all the RNAs are simply a polynucleotide. Nucleotides strung together into a polynucleotide. But unlike DNA, RNA, number one, is a single polynucleotide. Number two, has ribose instead of deoxyribose, and you can't see that in this image, but that's where the R in RNA comes from, ribonucleic acid, where the D in DNA comes from deoxyribonucleic acid. 3 has uracil instead of thymine. That is pointed out in the diagram. And the 3 main types of RNA, as I already showed you on the previous slide: messenger RNA, it's abbreviated mRNA, transfer RNA, abbreviated tRNA, and ribosomal RNA, abbreviated rRNA. And you're already familiar with these and working through the lab, I'm sure. Each group of 3 nucleotides in messenger RNA is known as a codon. So every triplet, nucleotide triplet here is a codon. So this is a codon, this is a codon, this is a codon, and we don't have a complete codon after that. Messenger RNA carries the message. That's why it's called messenger. The sequence, the message is encoded in the sequence of nucleotides, though. That's the information. But it's the message of the gene that's to be translated into a polypeptide or protein. So that's specific for messenger RNA. Ribosomes are the smallest non-membrane bound organelle that are found in all cells, including both prokaryotic and eukaryotic cells. And their function is that's the site, that's where translation happens, where proteins are made from the 3 RNAs. And the ribosome itself contains one of the RNAs. It contains ribosomal RNA. So part of the ribosome is made up of ribosomal RNA along with about 80 different other proteins. That's what makes up a ribosome. You'll notice too that there are 2 subunits. There's a small subunit and there's a larger subunit, and both of them have RNA, ribosomal RNA. And that ribosomal RNA functions by interacting with both messenger RNA and transfer RNAs. And the small unit basically is going to interact with messenger RNA, and the larger subunit is going to interact with transfer RNAs. Speaking of transfer RNAs, tRNA, and it's kind of coincidental that tRNA folds itself up into kind of a T-shaped. It's also sometimes described as a hairpin. I don't know how that started, but you see it a lot in the literature. One end of the tRNA is bound to a specific amino acid. So this is just a very generally labeled amino acid here. It could be any of the 20 different amino acids. The other end has the anticodon. And again, it's a nucleotide triplet. It's 3 nucleotides. And as the name implies, the anticodon is complementary to the codon in messenger RNA. It's going to match up with the codon in messenger RNA. So it interacts with both ribosomal RNA. It's going to bind to the ribosomal RNA that's in the large subunit of the ribosome, and the messenger RNA, in that its anticodon matches up with the codon. All 3 RNAs are made the same way in the same place. They're all made in the nucleus. Ribosomal RNA, though, more specifically, is made in the nucleolus of the nucleus. It's kind of the nucleus of the nucleus. That's the way you can think of it anyway. And that's where ribosomal RNA is transcribed. But this is the process of transcription. It's RNA transcription, and all 3 RNAs are made this way. So there are separate genes for messenger RNA, transfer RNA, and ribosomal RNA. And that's what I was referring to when I said that a gene is a sequence of DNA that codes for a protein, but it can also be a sequence of DNA that codes for a functional RNA. And transfer RNA and ribosomal RNA are functional RNAs, and there are genes that code for them. So a lot just came up, but it's basically just describing what's going on here in transcription. So this big tan sphere here is representing RNA polymerase, and it is an enzyme. You know it's an enzyme because it ends in ASE, and it's an enzyme that polymerizes RNA. In other words, it takes RNA nucleotides and strings them together into a polymer. So it's a polynucleotide, an RNA polynucleotide. And the sequence in that RNA polynucleotide comes from the sequence of a template strand in DNA. So the RNA polymerase unwinds and unzips the DNA and adds RNA nucleotides, complementary, that match up according to the base pairing rule, to the nucleotides on that open strand, that open template strand of DNA. And then when it's done doing that, once it has made the RNA leaves, and the DNA will zip back up again. So that, in a nutshell, is transcription. RNA polymerase unwinds and unzips the DNA, adds nucleotides, matches up nucleotides to the DNA nucleotides on the template strand. As the RNA polynucleotide is made, it leaves, and the DNA zips back up again. After the RNA is made, it undergoes what's known as RNA processing. So the RNA specifically the messenger RNA that comes from transcription is referred to as pre-messenger RNA. So that's what we're seeing here. So here's the DNA, and the DNA is being transcribed into RNA, messenger RNA, and then the messenger RNA, the pre-messenger RNA, goes through processing and then becomes the final messenger RNA. So that's an overview of the process of RNA processing here. So the pre-mRNA is the mRNA after it has been transcribed, but before being processed. That's just a definition of the pre-mRNA. You'll notice that in both the DNA and the pre-mRNA, there are regions, there are sequences labeled exon and intron. So these regions that are in the DNA get transcribed into complementary regions in RNA. But these tan ones are the exons, just like the blue ones in the DNA. The darker blue ones are the exons in the DNA, and the tan ones are the exons in the pre-mRNA. The light blue are the introns in DNA, and they kept them light blue in the diagram. And so what's happening here is the introns are cut out, and the exons are spliced together. So the introns are non-coding regions, referred to as non-coding regions of both the DNA and the messenger RNA. And because they're non-coding, they get cut out. The coding regions, the exons, get spliced together, and they're the nucleotide sequences that are actually going to code for the proper sequence of amino acids in a protein. So this is our final messenger RNA. And to top it all off, a cap and a tail gets added to the messenger RNA, both to enable it to leave the nucleus or to help it leave the nucleus through the nuclear pores in the nuclear envelope, and to protect it from degradation, because there are enzymes waiting for messenger RNA out in the cytoplasm that will break it down, make it go away. So how does this happen? Well, of course, just like most chemical reactions, or all chemical reactions in living things, it is regulated by enzymes. Enzymes run the show. Those proteins that make all of metabolism happen. All chemical reactions in living things happen. So this is just what I was saying about the cap and tail, and how the cap and tail help the messenger RNA both survive and get out of the nucleus. And then so you might think to yourself, well, why does this happen? And one explanation has to do with what was discovered by the Human Genome Project. You may or may not be familiar with the Human Genome Project because it happened a while ago, but that was a project where they sequenced the DNA of a human to find all the A's and T's and G's and C's that go into that making a human. And they found that humans have about 25,000 genes, which is a surprise because they thought it would be a lot more than that. And part of the reason it was a surprise is because there's about 100,000 different proteins that make up a human being, but there are only 25,000 genes. So how the heck can we get 100,000 proteins from 25,000 genes if one gene makes one protein? That doesn't match up. Well, this partially explains possibly why. These exons can be spliced together in a variety of ways. So, for example, this exon and this exon could get spliced together while this exon gets thrown away. And the same thing can happen in a different combination. Or this exon can be spliced to this end of this exon, and then these two splice together. So they're just spliced together in a different order. And when you do that, a different polypeptide chain is going to be produced from that messenger RNA. Another reason is that in the DNA, it allows for what's known as exon shuffling, and new genes can arise by exon shuffling. I'm not going to go into great detail about that, but that's another possible explanation for why these introns and exons exist, requiring DNA processing after transcription.